My name is Dr. Bromberg, and we're going to talk about variation in protein space. Okay, so we're going to start out with some stuff that you may or may not have read. It's been translated to over 400 languages, the book, Master and Margarita, by the Russian master Mikhail Bulgakov. And this is just a small piece, and it talks about this regular guy coming to the devil and wanting something from the devil. And the devil is not having any of that. He just says, when are you going to die? The devil wants to know if the guy knows when he's going to die. Makes total sense to me. The guy says nobody knows and it's nobody's concern. And of course, one of the obliging devil servants says, sure, nobody knows. He'll die in 9 months, next February, of liver cancer in the clinic of First Moscow State University in ward number 4. And why do I bring this up? Very simple, because the one thing we want to be able to do, the holy grail of genomics, is figure out when and how somebody will die. Well, hopefully, maybe not how somebody will die, but what disease will they have and how can we prevent it, okay? So that's the idea behind everything that we do. But in order to be able to predict disease, the first thing that we need to answer is, what is disease? Does anybody know what disease is? Seriously, does anybody know what disease is? Talk to me. Yeah?
Malfunctioning of something that should function properly.
Excellent. So you would be very surprised how many people cannot define it this way. They'll give examples, right? So this is perfect. And if you look at the Merriam-Webster dictionary, which I would argue is not the defining authority, but close to it, a disease is a condition of the living animal or plant body or one of its parts that impairs normal functioning and is typically manifested by distinguishing signs and symptoms. Okay? So there is a few things that are important here. The most important thing is that you have to have a baseline normal, right? Something that happens under normal circumstances. And these things are going to be different in Munich versus sub-Saharan Africa. And this is going to be a very different subset of measurements of the conditions of the living body which are considered normal, okay? Another thing that you need to know in order to justify disease is, how do you change? Which direction? Do you go up or down? And then you need to also know by how much, right? So what is considered disease? Because between you and I, there is lots of variation. So is any one of us sick? No. We just have different normal baselines. So there is some amount that needs to be defined in order for something to be defined as disease. So let me give you an example. This is the simplest pathway in your body. Arguably, maybe there are simpler ones, but this one is pretty simple. And it's also the most basic. It's the folate metabolism pathway. And the folate metabolism pathway, the malfunction of the folate metabolism pathway, is going to give you everything from lung cancer to heart disease, okay? Everything. You can think of a disease, you can get it. And in fact, they tell pregnant women to take lots of folic acid so the babies are healthy. You need the folate metabolism pathway to function well, okay? And so there is two enzymes, there is one enzyme in there that could be activated either directly from folic acid or from dietary folate. It's called DHFR, dihydrofolate reductase, okay? So DHFR is a protein, and let's say we were going to knock it out to make sure that the pathway malfunctions, right? So this is doing it on its own. My computer decided that I already talked enough about this and switching slides, great. Okay, so let's say we wanted to knock it out. So what is DHFR? What do you think? It's a protein, but what's the general term? It takes folate and it converts it into dihydrofolate or tetrahydrofolate. So what is this type of protein? It's an enzyme, right. So enzyme is one type of protein, but just so you are aware, because we usually talk about enzymes because they're the vast variety of enzymes in the body, but just so you're aware, there's antibodies, contractile proteins, transport proteins, regulatory proteins, structure and storage proteins, and you can talk about all of the functions of all of those, okay? So we're going to focus on enzyme just because it's easy to talk about. So that enzyme over there is DHFR. Okay? What do you know about its function from its structure? Anybody? Nothing. Great, me neither. I have no idea about anything about this protein from its structure other than it looks weird. But nice, I like it. All right, so this is the sequence. Anything you know from the sequence? What can I do with the sequence? Yeah?
You can try to predict, first of all, the secondary structure if I don't have the three-dimensional structure, but also maybe make predictions about whether it's found in the membrane or, in this case, since we know it's an enzyme, probably not, or in rather hydrophilic or soluble.
So you can make a lot of predictions with sequence, right? You could also make a lot of predictions with structure. Right? So that's important to remember. So if you have the sequence and the structure, you're already in much better shape for this particular protein than for many, many of the other ones that you have, right? So the idea with the sequence is that you, of course, can do multiple sequence alignments to find orthologs or proteins in other organisms that are related to this particular protein, right? So you can somehow extrapolate functional similarity in some cases, not always. And you could also look at, you know, prediction sites, right? So from alignments, you can say whether it's a binding site or a post-translational modification site and so on and so forth. So what I know right now is that there are these 4 positions from experimental evidence. I know this. There are these 4 positions, the L23, the leucine in position 23, the phenylalanine in position 32, the glutamic acid in position 36, and the, glutamate in position 36, and then the asparagine in position 65, right? And if I mutate those positions to the other amino acids that I've listed up there, I am going to have a really large number of experimentally measurable effects, right? So we have decreases in affinity for NADP and dihydrofolate over 10-fold, decreased affinity for methotrexate, and catalytic site activity is decreased. You can have reduced catalytic rate 5-fold, 3-fold, 2-fold, and yeah.
How do you figure out this effect?
This is experimental. So someone went in and measured experimentally how this enzyme functions with mutated positions.
No, but I get it, but how do you measure it? Have you, like, I don't know, lab animals tested?
You express the protein and do it in vitro, right? So you don't need lab animals. You can do it in a beaker, yeah. Right? So in order to figure out how much enzyme activity exists, what you do is you put in the components that are necessary, the input, and then you measure the output after a certain period of time. And you can do this for mutants versus wild type, and you can say, okay, it takes longer to get to 50% concentration of output. Yeah. Okay? Makes sense, right? So I can do this experimentally, no lab animals required, although lab animals would have been a fair game in this case. Although here in computational biology, one of my major motivations is to reduce the use of lab animals as much as possible. Okay, so you can do all of this stuff. Any ideas? You know, remember, this is the sequence, where they are in sequence. Any idea where these residues are in structure? Remember, we're talking about decreased catalytic activity and decreased binding activity. What do you think? Can you try to identify a binding site here? Just from looking at it. Sorry? So the one thing that I can let you sort of in on, and don't quote me on this, although this is being recorded. Most proteins look like Pac-Man. Those of you old enough to have played Pac-Man, right? This yellow thing that runs around, eats all the pills around? Yeah, okay. So most proteins look like that. Can you find the mouth? And that would be your binding site, okay? Now, this is a very rough approximation, so don't really do this consistently. But in principle, that right there is a binding site, right? So that sort of entryway into the protein. And guess what? These positions that I was talking about before, the N65, Q35, L32, and F32, are right there. They're right next to the binding site. So it makes total sense that by mutating these residues, you are going to affect binding. Okay? Just physically. So this is the binding site, And this is what a ligand would look like inside the binding site, okay? So we have that. But now we started talking about this disease story, right? We don't know that these mutations that decrease the protein function or change the protein function are actually involved in disease. We don't know this. They may or may not be, but we don't know this, right? What we do know is that there are two other mutations, L80 in position F and D153 mutated to V, which actually are involved in disease, right? But we know nothing about what they do for functional activity. And guess what? These positions are actually fairly far away from the binding site, right? So they're disrupting function somehow else, okay? So the main message here that I would like you to remember, other than the fact that those mutations are associated with megaloblastic anemia, right? So they are anemia. Oh, sorry, one message here is that when you read disease descriptions, correlation of mutations with disease, you get this stuff, right? Ranging from severe developmental delay and generalized seizures in infancy to childhood absence epilepsy with learning difficulties to no symptoms, okay? How can you have a disease with no symptoms? That's a very interesting question to remember, okay? In some cases, that would not be a disease, right? So disease, the main message there was the disease is not equal to disruption of function. It doesn't necessarily need to be the same thing, okay? Okay, so, but then we also talked about structure-function relationship. Remember how I said, binding site, blah, blah, blah. So here's the protein that we were looking at, and this mutation, L23R, that decreases affinity for an ADP and dihydrofolate over 10-fold. That thing is pointing to that leucine in position 23. What do you think is going to happen to structure? So if I mutate that position, it will change. Did it change much? It didn't. So it's not necessary for structure to change, for function to change, okay? That's a very important message to remember for you guys. And here's one more thing. Those of you who are somewhat more biologically inclined know that fitness, right, fitness of an organism is something different from structure and from disease, right? And unfortunately, we can't really measure fitness of humans, although it would be nice. But we can measure the fitness of E. coli, right? And E. coli also has this pathway, also has this protein. And this protein looks not very different from the human protein. That's the E. coli protein. Has slightly different amino acids there. But if you point to the same site, you can do experimental measurements of the fitness of this particular protein, of the E. coli in the presence of mutated protein. And this would be no lab animals, but E. coli, So in the end, what you get is a couple of measurements. So I'm just highlighting some of them. This is the catalytic efficiency, which is function, right, of the protein, and drug resistance, which is fitness of E. coli, right? So obviously, if you introduce a drug, you want to survive that drug, so the more fit you are, the better. So what we see very interestingly here is these, for three mutations, L28R, A26T, and P21L, the left side, the blue bars are wild type, right? And everything to the right, the three bars are mutations. So what you see over here is that you can have increased catalytic efficiency as compared to wild type, but absolutely the same drug resistance, right? No point there. And on the other hand, you have decreased catalytic efficiencies and increased drug resistance there as well as there. So functional effect, right, can be very different. So function may have an effect on fitness, functional change may have an effect on fitness, or it may have a reverse effect on fitness, and obviously it can have a correlated effect on fitness, right? So fitness and function are not the same thing either, okay? And actually, if you look at it in this sort of Venn-ish diagram, and these are not to scale, okay, but somehow, more or less, how I imagine this life to be, you have the space of variance. Everything that's white in the black box is no effect. The variant has no effect. Everything that's blue has an effect on function. And then everything that's yellow has an effect on fitness. And then a tiny little portion of these is a disease variance, okay? But what you see is that even though they overlap, there is a difference between them. There are things that have an effect on fitness that don't change function. How is that possible? Okay, what about those disease variants which have no effect on function? How is that possible? And this is not a straightforward question, so any guesses are welcome. Anybody? Okay, so I've had time to think about, so I can give you an answer. So these things, the yellow ones that are not in the functional end, their effect, let's say, through protein quantity. So if I evaluate how well a particular molecule of a protein functions, it doesn't mean that the same number of proteins is going to function the same way, right? So if I have one protein, it could be more or less functional. One molecule of a protein could be more or less functional. But if I have fewer of these proteins, it doesn't matter that they're fully functional. Does that make sense? Okay, that's one. And then this one, I think, actually only is very interesting in humans. We are a very selfish species. We talk about disease in humans mostly, and in animals that are interesting to us. We don't really care about disease in E. coli, for instance, if there is such a thing. But the idea of here is that you could have disease post-reproductive age, at which point you are no longer interesting from the fitness perspective. Right? So once you've had your children in the species world, you are no longer interesting in the fitness perspective, okay? So disease post-reproductive age has this ability to transition over generations because it doesn't prevent you from having children. Okay, so those are the things to remember. So this is back to the question of how do I know that the function is disrupted? Usually do some experiments. Yeah?
Could you elaborate a little bit what we talk about when we talk about fitness in E. coli? Is it like how much of them reproduce?
So E. coli fitness is usually measured in the number of cells, yes. So how many are after a certain time? How many are there after a certain time? And with drug resistance, what you also do is you put the drug into the environment and you see how many cells are there in wild type version of E. coli or versus the mutated version of E. coli. Yeah?
So in a biological sense, like, maybe on a high level, is it right to describe fitness as a measure on how well you or a species can reproduce?
Yeah. So fitness is really about reproduction, yes?
Coming back to that space of variance, are these mutation variants? Different mutations that are possible or what?
Yes, and so we will elaborate on this in 2 seconds. All right, so the first thing is, this stuff is called the traffic light experiment, right? So depending on the color, you can say that something is or isn't whatever you are measuring, right? Okay. But unfortunately, or fortunately for us, depending on how much you like beakers and flasks, we do computational stuff. And this is another way to figure out whether function is disrupted or not. SNAP2 is very dear to my heart. My PhD thesis was on SNAP1, so it's nice. But the idea is you submit a protein sequence and we make a prediction for you. Okay, so aside on SNPs. SNPs are single nucleotide polymorphisms, you know, say the single mutation of the DNA sequence, substitution, and 99% of the variation in the genome is SNPs. And they're all going to be single nucleotide polymorphisms. But not all SNPs are going to be in the coding regions. In fact, there are significantly more SNPs in non-coding regions, just trivially, because there are more non-coding regions. And not all SNPs in the coding region are going to have an effect. But so when we talk about mutation in this class, we're going to talk about missense mutations, non-synonymous coding region mutations that cause an amino acid substitution in the protein sequence. So obviously it's a sort of a narrow way of looking at mutations, but this is the most frequent type of mutation of the coding region mutations, okay? So keep that in mind, and we're going to talk about that. So what are the 3 things that are important if we're going to build a computational method for predicting functional effects of mutations? What are the 3 things that are important in order to predict effect of the mutation? So the first thing we talked about very extensively was the structure story. What is it? Yes, how close are you to the binding site? So that's going to have an effect, right? So if we are looking at this DHFR L23, this thing is right there, and if, let's say I was trying to make prediction for functional effect of DHFR L23R, I would say, you know, is it close to the binding site? If we had this information, we can say this. And in this case, yes, it is close to the binding site. Never mind the fact that you don't always have this information about the protein, right? And the word close is a very strange word, because what is close, right? So the idea is that you have to set some thresholds, and I'm sort of sweeping that under the rug. Okay, so you've got this. And then the next question is, we talked about this as well. Next question is, are there any structural effects? Right, so you may not be close to the binding site, but you can ask, are there any structural effects? Does the protein misfold, right? And in this particular case, I told you that there is no structural effect. And that usually happens, or often happens, not usually, with amino acids that are close to the protein surface, right? So the idea is that if you are not really inside the protein, if I substitute you, you're not going to be a problem, okay? So then in this particular case, no, that's not a problem, okay? And again, you have to discuss what is and isn't a problem in structural terms. And finally, the one thing that we sort of hinted upon but never mentioned is this idea of other proteins or related proteins in a similar species, right? And their alignments. So you guys know what conservation is, right? In multiple sequence alignment. You have learned this at this point? Okay, so this is a really poor resolution representation of as multiple sequence alignment. Are you familiar with these logos or no? Yeah, okay. So what we're interested in is this position number 23, and the mutation is L23R. So what do you think here? Is this a problem or not?
Yes.
Okay.
Relatively, L is more likely.
Right. So the majority of the proteins are going to have L in that position. So probably this is a problem. But there is something really important to think about here. You see that I and M and V are also fairly large. So actually this position is tolerant of mutations, right? So maybe it's not as much a problem as we know, okay? And then, so there is this other version of conservation, which most people don't talk about. And this is just a basic biochemical conservation. Every amino acid has its own properties. And given its properties, you have to somehow try to figure out whether this mutation is going to be an issue. So, for instance, here the leucine is an aliphatic amino acid, whereas the arginine here is positively charged polar amino acid. So, you know, there may be an issue. Actually, the rule of thumb is every time you substitute in or out things that are charged, you're going to have an issue, right? That's a rule of thumb. But that's clearly not going to be applicable anywhere, everywhere, so keep this in mind, okay? All right, so this is also a question mark. So in the end, we have all of these three things, and I can tell you that yes, we are close to the functional site. No, there is no structural effect, and I'm not sure about this conservation story. So now that I told you this, are you any closer to figuring out how this mutation is going to affect function? No. And this is how I feel about most research. You do all of these things, and then you're like, okay, so now what? So now what? You catch somebody, open their brain, throw the things in there, and get an answer. Unfortunately, we're not allowed to do that. That would not be lab animals. So what we're going to do is some AI, okay? And I know that for this course, this may be a little basic, but just to sort of run through it anyway, if you imagine the axes, the structural effects, the active site proximity, and conservation, you can, of course, map every single point onto this space, right? And then what you can do is you can say, okay, if I have done some experiments like I have, and now I know what the answer is, I can label it, right? So I can train the model in this way, and I can try to separate the answers, okay? So again, just to sort of be very clear, let's say we have this point A, which is blue, and we have this point B, which is red. How do we separate it? Any way that separates them, right? So this is one way. Let's say I had another point, I would have to adjust, and I can do this all the time. And of course, this is my favorite. Anybody know how to do this? It's not linear, all right? So we can do this. So the idea is very simple. And then from here, what I can do is I can make predictions, right? So I can take a thing for which I don't have a label, this thing, for instance, and ask what color is it. And for some reason, it switched colors. So the outlines are red instead of the actual filling is red. But the idea is that this black thing is in the blue range, right? So the prediction is blue. And this one is also blue, right? So what is it that I can also know from this prediction? Anybody? Other than blue, other than one answer blue, what can I learn?
In this case, also something about the distribution, maybe?
The distribution of what?
I mean, since you basically map it to a certain space, you can actually see which space is rather filled or relevant by red and blue dots, so you can actually kind of try to understand what the criteria would be in blue or red.
It's an interesting concept because it sort of halfway answers my question and half goes into something else entirely, which is nice. I appreciate that. That's figuring out which features are important for making the prediction, right? But what I was referring to is this distance to the separating border, right? And yeah?
Just a technical question. Maybe I missed something, but why everything is blue?
Yeah, it's not blue. So the problem is that when I transferred the slide, it put the outlines in red instead of filling in red.
So half of them imaginary.
So the thing in the middle has red outlines, which you cannot see because of the resolution, and they don't display well. Yeah, okay, so the thing in the middle is blue. I apologize for that, but they were red in the bigger circle thing, yeah.
And another question, why do you have the blue line that goes from the origin to...
Why are there two separating?
No, like the blue line that's in the middle.
Oh, that's an axis.
Okay.
Sorry. Okay. So then the idea is that you can actually define reliability. And this is going back to the original question that I asked you. How does the reliability relate? Okay? All right. So SNAP was a neural network-based method. You guys know what neural networks are, right? Okay. SNAP used a bunch of features and it produced two outputs. Yes and no. Okay? Really nothing major. Yes, functional effect. No functional effect. Anyone see a problem with that? Yeah?
Sounds kind of hard to define.
Yeah, there is this whole threshold thing, right, about what is a yes effect and a no effect, right? And so when you have a stoplight experiment, when this thing is red, this thing is yellow, this thing is green, you can do that. But in most cases, you don't have that experiment. So there is some kind of a fold. So is 5-fold change in activity a yes effect? Is 2-fold change in activity a yes effect? 1.5-fold? And this is how it becomes an issue, right? So keep this in mind as we go on. So this is how SNAP performed versus other methods. Have you seen curves like this before? You have, right? ROC curves. Yeah, okay, so you know that you have to be higher, right? That's the idea. So versus Snap, the version that Snap 2 outperformed the earlier version, yay, no. But it did better than SIFT and Polyfan 2. And if those names don't give you anything, I can show you how many methods there are for prediction of functional effects out there right now. Okay? There are at least 88. And you know what we know about methods that, you know, when there is a lot of methods out there to do something? Why do we have a lot of methods to do something? Because nobody has answered it, right? So no one method does well. That's why you have a lot of methods. So then, what's the difference between these methods? And you can tell, we put together not 88, but 10 or more or so of the ones that were used commonly. This method just uses conservation. This method uses just structure. The methods in the middle, including Snap 2, use many features. And then you have, you know, the next stage, which is many methods, right? We'll just put many methods together and try to figure them out. And in the end, you get a ROC curve for every one of them. And everybody does better than everybody else. This is the most amazing story that I've ever seen. Everybody does better than everybody else. No matter what, because otherwise you don't publish a paper. What's the point, right? But it's really funny how, you know, everybody does better than everybody else. But in the end, that just means one thing. Nobody does well. Okay? All right, so, and they all do about the same. And as I told you before, there's methods that have one feature, methods that have many features, methods that have many methods. So why do they all do the same? And one of the reasons is because evolutionary conservation is a driving factor in most cases. So nature has had 4.2 billion years since the evolution of life, right, to try to estimate what is the best residue to have in this particular position. It was this worldwide thing of making, you know, experiments. We're gonna do this, we're gonna do that. And it tried everything, really, it tried everything. So the things that are conserved are the things that work. That's all. And if you recall that chart that I showed you, all the things overlap. So function, disease, fitness, they all overlap. So really we're talking about a small set of variants which are different between fitness and function, disease and function, and so on and so forth. So when it comes down to us, you know, the people that have been trying to do this for the past, I don't know, 20 years, maybe 30 years, right? Trying to figure out what nature had done over this span of time. We only have very limited data sets. We look at very few things, right? And because we're limited in data, we are trying to use this data creatively to make predictions in all of these three different fields, right? So the pathogenicity, impact on function and structure, and evolutionary fitness from the same data sets, okay? So this is what happens. We take the same features, the same data sets, and try to predict three different things. Awesome, right? How often do you think that works? Obviously, it doesn't, right? So the idea is that it works broadly because of this overlap, right? But it doesn't work for these particular things where you are trying to focus on specifics, okay? And in fact, we know on a larger scale here, the red and the blue lines, they represent disease variants, and everything else, all other variants are represented by black and, the red and the green are disease variants, the black and the blue are everything. And what you see here is that most disease variants happen to be in conserved regions, right? So disease variants are conserved, except that they also happen to be in non-conserved regions, right? And then there is also these pesky guys who are conserved and have no effect on disease. Happens, right? And then there is the story about the structure. So disease variants are frequently destabilize the protein. So this burgundy colored, purple colored pie is the disease, of the disease variants, that many destabilize the protein, plus this, you know, whatever that is, God, the 7% pie, they destabilize the protein as well, right? And they have an effect on function. Sure, so, but then, you know, there's that blue piece, which are disease variants that don't destabilize a protein, and we've seen one of those, right? And then there is also the problem with the all variants in the non-disease data set. Apparently there is a whole bunch of variants that are not disease related and still destabilize the protein, so that doesn't actually correlate very well. So in any case, what I'm trying to tell you is that you need to know what it is that you're trying to predict, right? Is it function? Is it structure? Is it disease? You have to figure out what this particular variant is that you're trying to look for, right? What is the effect you're looking for? Once you know this, we have different set of problems, all right? Never gets easy. So the problems that we have is this. You can imagine that whatever effect you're looking for, you can separate the space and the feature scale of effect and no effect, okay? So this is how these variants look. To be a little more precise, I can always tell you when there is an effect, right? So, are there any cars in the world? Any red cars in the world? Yeah? Yeah, there is one right there. Yes, there are red cars in the world. I can tell you when there is an effect. There are red cars in the world. Are there any yellow cars in the world? I don't know. Let me check. Yellow cars? Yellow cars? Still no. At what point do I stop? Right? And unless I've exhausted every single possibility, I don't know at the end of this experiment that there are no yellow cars in the world. So, at the end, I still don't know, and then this is where this distribution comes in handy. From a very good collaborator of mine. This is effect, no effect, but in the reality you have effect and unlabeled, right? So you don't know whether there is no effect. You just know that there is a set of variants for which we know there is an effect, and there is a set of variants for which we cannot find the effect, or we haven't looked for an effect, okay? So this is positive unlabeled, and what you want to do is use this, yeah?
Could you please explain a bit how to understand the x-axis here? So what is on the x-axis?
It's a feature scale, whatever feature you're looking at.
So for one particular feature?
Well, so I'm sort of abstracting away into this feature which actually defines whether this thing has an effect or not, right? So in the end, what happens is you have a yellow distribution that's on the right of the things that you're observing. Right? And you know that underlying it is a distribution of effect and no effect variance. But you don't know what that distribution is. Okay? So what they try to do is to figure out what is the prior probability of it being effect or no effect. Right? And this is what define these sub-distributions of the underlying the yellow distribution. And actually, if anybody is interested, these are the papers to read. Okay, we can actually do this, and this would be the right way to train the methods, except who knows how many methods are trained this way? Huh? Anybody can guess? Yes. It is one very good point to note here is these papers are 2016. I don't think there have been enough time for us to use this stuff yet. So none of the current tools are positive unlabeled, right? Or not that I know of just yet. Okay, so the most common way to train these things is to use OMIM data. OMIM data is disease variants which have monogenic disease, right? So if something is monogenic disease, chances are this thing affects function, this mutation affects function, right? Because you have one gene that's affecting disease. Okay, and most people will set that equal to disease, and then they will take evolutionary experiments, right? So take a multiple sequence alignment and orthologs and say that anything that differs between orthologs is probably neutral, has no effect on function. In both cases, these are approximations. Right? So you can have variants that do have an effect on function and that don't have an effect on function, but you could also have variants that are across species, so let's say a E. coli DHFR will probably not function at the same rate as the human DHFR, right? So the fact that you could substitute variants doesn't really mean that there is no functional effect, right? But this is what most people use. We didn't use this set. We also didn't use positive unlabeled, but we used a very different data set for training SNAP. And that data set is actually experimentally determined functional effects. So not this approximation of disease versus orthodox, but actually experimentally determined things. And this allows us to do some computational experiments which we couldn't do with things that have been trained on this maybe data, okay? So there's one thing that we did. So first, before I go into what that thing was, this is just an understanding of how SNAP scoring works. So again, remember, SNAP has this 2 output nodes that are 0 to 100, and you compute the difference between them, right, to make the prediction. So the SNAP score is minus 100 to 100. And ideally, in an ideal world, you would have minus 100 neutral and plus 100 non-neutral, right? And then usually, because it's neural networks and you know, kind of are like them, we're not very ideal, so we kind of go in this space, right? So anything under 0 is neutral, and anything above 0 is non-neutral, okay? So then I asked, how well do we perform in identifying variant effects? The thing on the right, the distribution on the right, with the shift on the right in red, is the variants which have been shown experimentally to have a very severe effect on function. Like, really, there is no question about it, there is an effect, okay? And then the things on the left are either these synonymous substitutions. Basically, you're not replacing the amino acid, you're putting in the same one, that's normal. And then these orthologous substitutions that I talked about, which are alignments across multiple sequence alignment, the same residue in different orthologs. So the differentiation is quite striking. I mean, we still make errors. So for instance, the things in dark blue, the ideal, the synonymous non-neutral, the synonymous mutations, they shouldn't have an effect, right? So we're taking a methionine in position 27, we're putting a methionine in, there shouldn't be an effect, right? But they do, it's predicted to have an effect, so it's a limitation, yeah.
How big is the data set here? Because at least the way it's plotted, I think there's quite a lot of dots in the long tails of curves that actually go into the wrong prediction direction.
No, so the data sets are plotted, they're the same way. So this is a fraction of variance, right? So this is not the total number of variants, it's a fraction. So the error rate here is about 20%, and the error rate here is about 10%.
Okay.
So that's an interesting observation. I would never have guessed that it's visible from here. But what you're doing is you're counting the area under the curve, not the...
Okay, yeah, okay, then
it's still higher.
Yeah.
It's still about twofold, more or less, 1.7-fold, yeah.
Maybe it's trivial that it is, but this is not test set, right? This is not the data that the algorithm was trained on.
This is the data that the algorithm was trained on. So this is just the limitation. That's what I'm trying to sort of indicate, right? The idea, actually, you can see that it's the data that the algorithm was trained on because it does better in predicting orthologous mutations than identifying that the same mutation is a problem, is not a problem, right? So the light blue curve is somehow shifted to the left. So this is the things that it's supposed to be doing really well on, okay? So this is as good as you're gonna get, and obviously those are prediction errors. So this is just as a side note, all methods are going to have prediction errors, even on the data that you train it on, right? It's not a very clear separation there. Okay, and so these are possible prediction errors, right? So you can argue that actually the method is picking up something in the biology which the experimentalist didn't see. Arguable, okay, but it is possible. But in any case, I call those prediction errors, okay? Fine, those are prediction errors. So now here is the interesting thing. Remember, SNAP was trained positive, negative, yes or no? And all of a sudden, I take the experimental data where I have the effect, I have severe effect, I have mild effect, and I have moderate effect. The other way, severe, moderate, mild, okay? And what I see is that snap reliability actually differentiates groups of variants, okay? Quite clearly. It moves the things according to severity to the left, towards the neutral side. It was never trained to do that. Think about why it's doing that, okay? Okay. Anybody know why it's doing that? No? All right.
There is some quantitative pattern of like your independent variables affecting dependent ones. Maybe this is not just 0 or 1, right? This is distributed among some continuous scale. So maybe your algorithm just captures it.
Yes, but how? If it's not trained to do that?
This is probabilistic in the end, right? So
where is the probability factor coming from?
Your output units. It's not spitting.
No. It's on the input side. The probability factor has to come in on the input side, because if it was just the output, right? So if I told you can give me, if I trained it, let's say, be anything above 0 to be non-neutral, then that's probability. But I trained it to be non-neutral is 100. So then
it's your target, but what it spits out is still between 0 and 100.
Why?
Because it's not so sure.
But there is a reason why it's not sure, and that's what I'm trying to get at. Why is it not sure?
Maybe because in the end some human labeled the effect in some way and he had to use
Yes, whenever there is a problem, it's always a human problem. It's never the machines, okay? Well, not yet, until they come to take over. Okay, so this is the reliability. Remember this thing? This is what you were pointing out. Yes, it's probabilistic. The reliability displays something. And let me illustrate this very quickly. Which mouse has lighter fur? The right one. Yay. So that's easy, right? That's called severe effect. Which mouse has lighter fur? Exactly, right? So it really depends on the lighting and where you're sitting. So different people would label these things differently. So I can name the variant as having an effect or not having an effect, depending on what thing I'm using, what evaluation I'm using, just simply looking at these things, right? So basically what that is reflecting to me what is the difference between people putting a threshold of what has an effect and what doesn't have an effect, right? That's the probability. Okay. And the reason is because I will train on the same variant with different answers. And so the machine learning can't do this, right? Okay. So where do the neutrals go? What did I tell you about the neutrals? No effect variance. Come on. What did I tell you about no effect variance? Can I tell them apart very easily? No. So where would they go? Remember that left side curve, the really left side curve? Those are the idealized variants, right? Those are the orthologous neutrals. This is where the neutrals go. Okay? So this is somebody telling me experimentally this variant is neutral, has no effect. Does it look neutral to you? Right. So it looks like it's slightly more mild than the mildest of the variants. Okay? So this is what the idealized neutrals look like, just to recall. So why is the real neutral looking differently? One of the reasons is variant selection is biased, right? So this is a quote from a collaborator of mine. He says, "We investigated the pathogenic role of the missense mutations found in the morbidly obese cohort by comparing the activity of the different mutated melanocortin-4 receptors to that of the wild-type receptor." Other than scientific mumbo-jumbo, what that basically says is pathogenic role, we are looking for pathogenic role in a morbidly obese cohort, right? And we're focused, we know this protein is a problem, we're looking for causes, okay? So, are they looking for neutral variants? No. Are they selecting people to be able to find neutral variants? No. So the problem is they start out with this thing. So what does the output look like? You have a bunch of graphs. Actually, the content of most of them is not important for my point. But the idea is a bunch of graphs shows that they found some variants which have an effect. Great that they set out to do this, and they found the variants that have an effect. But then almost as an afterthought, in the bottom panel over here, they say, oh, actually, there's 2 more variants, right, in this particular thing that have the same wild-type activity of agonist binding on the y-axis as the wild-type. So they don't change function. So by the way, these 2 are neutral. That's how they identify them, right? But they identified them from a group of morbidly obese people where they were looking for a problem. So I am not sure about the neutrality of these variants, okay? And the funny thing is, a couple of years later, they do an experiment on some other activity of this protein, right? So this protein doesn't have only one activity. It has a couple, right? So here they're looking agonist binding. Here they're looking at constitutive activity. And what they find is this threonine 11 serine and arginine 18 cysteine, which they had labeled as neutral previously, are actually not neutral when you look at constitutive activity. Okay? So is this variant neutral or not? Well, we can assume it's neutral with respect to agonist binding. They measured it. But with respect to constitutive activity, it's not. So this variant is not neutral. Okay? So how does a neural network recognize this? Well, I give it 2 things to look at, right? And picks one, somehow in the middle. And then the other thing that's also important is that effect identification is subjective. So if I looked at the effect where the arrow points to neutral, yeah, everything is neutral. And if I look at the concentration where it says non-neutral, well, then there is a lot of non-neutral variants. So you also have to define your environment in which you're measuring your time that you spend looking and so on and so forth. So again, functionally non-neutral variants in our database actually happen to fall into these categories where half of them also disrupt structure and destabilize the protein and actually even cause disease. So can we make sure that we say that these effects are functional or not? Can we say that we are correct? I don't know. And this signal is exactly what the neural network is picking up. The neural network is identifying these not sure signals as severity of effect. Okay? All right. So the one thing that we also noted, so remember this is what the neutrals look like. The red line is what the severes look like. So there's still some difference between neutrals and severes. So obviously severes are more clear in terms of identification. So when we looked at the disease variants, identified disease variants, they're the light red ones that distribute very much like the severe effect variants. So that's, at least we're right on that. So most of the time, disease variants will have a functional effect. On the other hand, the things that we label polymorphisms, these are the things where we think that there is no disease association. They distribute kind of like these neutrals that we knew. So some of them will be neutral, and some of them will not be neutral. And so the take-home point here is that, when you walk around with a bunch of variants, 10,000 coding variants per every individual as compared to reference, you have 10,000 differences from reference. Half of those are not neutral. They define you as you. They define you the way you look, the way you act, and, well, maybe that's social. But the way you look, the way you feel, and what diseases you get. That's the definition, okay? And again, still dependent on the environment. But to sort of proceed from this world, I want to point out another thing. Most methods out there look at effect as on or off, right? It's either yes or no. But we live in a continuous world. I mean, it's obvious that it's not on or off. And what we found is that actually, if you recall that, these are the disease variants that are super conserved, and then, if you try to figure out which variants are not conserved in orthologs and call that neutral, right? And if you try to differentiate those as on or off, what you're doing is basically trying to differentiate conservation from not conservation in a very roundabout manner. right, in a manner that's not going to give you anything good. And of course there is error, you know, on that side, and that error can be very much attributed to this functional distribution. It's not on or off, it's a variation, okay? And so when we look on the neutral side, we also see, in the worst case scenario, what you're seeing here, this corner of the yellow is a huge, huge 24 million variant set of differences in multiple sequence alignment of 100 closest vertebrate relatives of the human, right? So these are supposedly neutral. And then you see that 313 of them are disease variants, right? Clearly disease variants, and actually probably even more than that, right? So what's okay for a chimp is not okay for us. What's okay for a dog is not okay for us, okay? And so this is how you know that the way that these methods are trained is incorrect. You can't do this. So the smallest estimate is that 3% of the variants which are found in other organisms are disease variants in humans, and actually up to 12%. So there is this ways that we can discuss why this happens. So they could be compensated, right? So you're looking at one variation in a protein, but you should be looking at correlated variations, for instance. But in the end, this is the estimates that we have. So what you should be talking about, and this is all my motivation for talking about this rheostatic behavior, where the on and off aren't flips, but rather versions of functional effects, okay? Okay, so to be sure that there are toggles, there are positions in a protein where no matter what you put in, you're going to have an effect, a really large effect, right? And then there are the rheostats, and these are the majority of the variants which can tolerate substitutions. And in fact, when you look at E. coli, this is the LAC1 repressor activity. Let's say the position tyrosine 47 in LAC1 repressor actually knocks out everything. Right? So all the measurements for all of the variants are pretty much up there. The Y47 is the wild type on the left, right? And you get at least 50-fold or 80-fold loss, but for the majority of these, you get a lot of loss of activity, right? And then on the other hand, the valine in position 52 has this range of functions. So it actually depends not on the substitution, but on the position in the protein, whether the effect of the mutation will have a toggle, yes or no result, or a functional result. And if you try to look at the distribution of the prediction scores of whatever method you were looking for, you would expect this sort of distribution, where anything below that middle 0.5, let's say, right, is neutral, and everything above is non-neutral. But note that for the things below this 0.5, you expect neutral to be neutral. You can't be more or less neutral, okay? So either rheostat or toggle positions, neutral should be the same. Whereas non-neutral could be complete, severe, or it could be a range, right? And this is what you're seeing over there. So the light colored is rheostat position, the dark colored is toggle positions, blue is neutral, red is non-neutral. And when you ask what do the methods do, it's quite disappointing, okay? So these are top, best performing methods out there. Really, really good methods. Not to put myself on the spot here, but yes. So there you go. You can see this is highlighted as the p-value. So what we're trying to differentiate is neutrals in toggle positions from non-neutrals in rheostat positions, right? So functional effect in rheostat positions is predicted to be less of a problem than neutral effect, no effect in toggle positions. It's a problem, okay? And then, this is incorrect, obviously, and then neutrals and non-neutrals in rheostat positions are poorly differentiated, right? So if you look at the rheostat position alone, the one that has this ability of moderating function, that problem of thresholding, which exists in the human world, also exists in the computer world. I cannot differentiate these very well. Neutral and non-neutral in the same type of positions, we don't do well. And in fact, we also don't do well for the toggle positions, so we can't differentiate on or off in the toggle positions either. Right? Which is very sad. So the one thing, and so also really interesting is that we do differentiate the neutrals in rheostat positions from non-neutrals in toggle positions, because apparently neutrals have a scale as well. Who knew? Okay, the one thing we are able to do correctly is to differentiate rheostat non-neutrals, the range functional effects, from the toggle non-neutrals. And the thing that we trained on, we're able to differentiate neutrals in rheostat positions, which is these multiple sequence alignment variants, from toggle non-neutrals, which are these positions where disease variants reside. And this is what we train our methods to do, and this is why we do so well. But for every other aspect, we fail. Okay? And this is actually quite important, because when you use these methods to make assumption statements about variance in human data, you sort of rely on their functionality to be correct, right? And taking this functionality into account, we can do some experiments which actually are super interesting. So one thing we found, for instance, is that the common variants in human populations, the things that you see around very much, are actually more functionally effective than the rare variants. It's counterintuitive, right? But we are used to thinking about function as disease, and this is not true. So you could be just different, right? And common variants are the ones that define ethnicities, for example. So ethnicities are different, and that's due to common variation, right? So there is a functional effect, but it's not a disease effect, okay? Is that clear for everybody? Okay. And then what we know also is that we can look at this expectation versus reality, and what you have here is 3 curves. The red curve is the mutations that are impossible via a single nucleotide polymorphism. So we see very few of those in real life. So, you know, you expect those to be deleterious. The blue line is where the mutations which are possible with a SNP. This is taking all mutations that you could potentially have. And then the white line here is basically saying this is the variance that we observe in the population. So you have the expected are the blue and the red. The white is the thing we observe, right? And the one thing that I can tell you here is that you can clearly separate the things into 3 fields. So the thing on the left is biochemically wild type and physiologically wild type variants, right? So the things that have no functional effect, fine. The things on the right have a functional effect. But at some point, if you look at this 23 where the blue intersects with the white, you see that the expectation becomes much higher than what we actually observe, right? And when the expectation becomes higher than the observation, you know that there is some natural process that's reducing the amount. And that's evolution. That's the fitness criteria. So basically, mutations which are functionally effective that you would expect to see if there was no selection pressure are removed by the evolutionary fitness criteria. Is that clear to everybody? Yes? Okay. So you could have an effect that has nothing to do with disease, nothing to do with fitness. It has an effect, and that's the differences between us that I've been talking about all along. So some conclusions. This is the end, more or less. Healthy people carry a lot of functionally significant variants. Everybody has roughly 45%, if you don't believe that number, a fifth is also fine, of the variants where there is a functional effect, some functional effect. Okay. Selection is restrained from removing many functionally non-neutral variants. You would say, okay, why is selection not removing all of these variants, right? Why are we still seeing something on the right? Well, because you want that. You know why you want the functional effect? Any ideas? Yeah?
Basically, by chance, you could also have a positive functional effect, and I suppose that's kind of the evolutionary.
That's kind of right, but there's a very tiny fraction of variance which is positive globally.
Yeah.
So if you follow that train of thought, you're positive environmentally specific. right?
Yeah.
Okay, so that logic is actually very good. So let's say lots of melanin in the skin, darker skin color, happens where? Where you need extra protection from the sun, right? And so that's beneficial in those populations, right? So evolution is not going to remove these variants from the population simply because it's beneficial for that population. Now, for northern populations where we are dependent on the sun to actually hit our skin in order to make some essential amino acids, right, we don't want to block it because there's little of it. So you reduce the amount of melanin. So the idea is that you want to be environmentally good, and evolution is not removing these functional variations because it's important. And, you know, God forbid some virus comes and wants to kill everybody. If we're all clones, guess what? We're dead. So there is a way to have something to work with in case of environmental stress, okay? So that's number one. Number two. And then finally, I think one of the more important things that we found here is that don't believe the wet lab. Don't believe the experimentalists. They are right a lot of the times, but they make mistakes. And all of their values are thresholded. They're calls. I believe this is the case, right? I believe this is the case. But when you train a machine to recognize the same stuff, maybe not, right? Okay, and finally, the one other thing that's important to remember is that what these variants we carry around, they can contribute to disease, and some diseases are more common in some ethnicities. So the variants that save you, the variants that make you different, the variants that are good for your environment are also the ones that make you have more chance of a particular disease. So it's a double-sided, it's two sides of the same coin, basically. Okay? So, and the most important conclusions for people who want to do this computationally, okay, fine. Evolution, as reflected in conservation, is not the only defining factor of the variant effect. Rare variation is not always causing disease and doesn't necessarily have to have functional significance. Common variation is not going to be functionally neutral. Functionally or structurally significant variants do not necessarily cause disease. Structural significance of variants doesn't mean functional significance of variants. And most current methods are not independent and thus give the same results. Okay? For all things you do in life, you should know exactly what you're doing. Okay? So if you ask these people that train these methods, 90% of them, you ask, what did you train your method to do? And they say, predict effect. Effect on what? You need to know what you're predicting, okay? So, the last straw broke the camel's back is obviously not the case, right? The back is broken before the straw lands. So, the bunch of variants you carry around in your genome predispose you to a disease, and then you have one more variant, or whatever, that actually gives you the effect, except there is no one more. It's everything all together. It's not that this variant gives you the disease. It's in the environment of all the other variants in your genome is that when you get a disease. Yeah, people who did work. Yes. Tell me.
Just a question, because basically over the course of the lecture we've talked a lot about how you use proteins across different species to compile profiles and thus reveal evolutionary conservation proteins. Now, since I think in the end most of the science that's done there has really the purpose to heal or predict human diseases and not, I don't know, cat diseases or whatever, the question is, first of all, is it possible, second, is it done that profiles are then compiled over different human variations of a protein to actually see what is relevant for humans across ethnicities or different populations?
So that's an excellent question. So now, for the past maybe 2 years, maybe 3 years, we have enough data to do this, right? We haven't had this data before. Remember, we only had one reference genome in 2001. right? That was the end of the Human Genome Project. So we've accumulated enough data to do this. People have looked at differences between ethnicities, but it's very hard to put a finger on what is the specific disease predisposition or specific functional effect of a particular variant across human populations. We're limited by a number of factors. One, you're not allowed to just randomly experiment on people, right? Yeah. So you have to have a hypothesis. Second, we don't have a large enough population to do this very, very well. So GWAS, genome-wide association studies for a particular trait, have been the rage for a very long time. So the idea is that you look at particular variants in people who have or have not the disease, or have not a trait, which is basically trying to figure this out. The problem is that if you think about the number of available variants in the human genome, and versus the number of people that you have available to measure this dependence, you're almost never statistically significant. So if you go to genome-wide association studies, first of all, you have to look at common variants. Because if you have a rare variant, you see it in one person in, you know, a million, that's it, right? But you have to have, let's say you have a common variant, and as I said, common variants are very rarely disease variants, right? So they're going to be functional, but they're not going to be disease, right, most of the time. And then you go to this population, you have 3 billion positions in the human genome, right? You have a variant about every 100 positions, 300 positions. So you have 100 million or so variants per person. What is it? Yeah, 100 million. So 9 minus 2 zeros, 10 million, sorry. You need to correct for that amount of testing every time you look for significance. And it's very hard. So when you go to genome-wide association studies that have been done, you find that the highest scoring SNPs, the highest scoring variants, will have odds ratios of 1.2 or 1.3, which is not something you can go with. So if you're familiar with this, let's say 23andMe, or all of these DTC, direct-to-consumer sequencing companies that will tell you you're predisposed to this, you're predisposed to that. They will do this, and then you go to another company, and they will tell you completely different numbers. So it's all really very vague. It's a great idea. Yeah?
Other questions. So basically the naive view on the whole issue was there are neutral ones, and there's some kind of variations which affect us very visibly. So basically, after your talk, I had the impression that there actually is no neutral one, but we don't know the effect yet.
That's actually a very strong argument in the field right now, that there is no neutral variation. So everything affects everything, right? To a certain extent. It's probably not true. Actually, very likely not true, because you will have a certain number of substitutions where, you know, we can't measure the effect at all. And you can argue that, you know, in the future we will be, but then the question becomes, is it reasonable that this level of change is somehow related to something that this protein does in the body? So if it's a 1.01% change in activity, this may be too low for even evolution to notice. So we have to threshold. At some point we have to say that 1 is 1, right? And how much is error in measurement, right? So you will always have some error in measurement. That's a very good observation, though. Anybody else? Yeah?
Technical question. So how you define exactly the variance? Are you taking like 1,000 people with the same mutation and check how many of them had the functional or undergone the functional change? And how
for this, or for this, I actually have a variant. So I sequence your genome and I see that this position has a SNP, as a nucleotide polymorphism.
Yes, but was it like, did it drive any functional change and did it drive any disease? So how is this checked experimentally? I mean,
so
the population.
How do you know that it causes disease? So most often the things we have are monogenic disorders. right? So this we infer this from monogenic disorders. Monogenic disorder is a disease which you can trace through pedigrees of individuals, where you can obviously see, you know, people that carry the gene are affected, those that don't carry the gene are not affected. So those are usually how it's done. When you get to multigenic disorders, it's much more complicated, clearly, but genome-wide association studies is one way of doing it. So you first find an association and then you follow up with experimental tests to see whether that changes. If you can build a good model for why this should work and you have a statistical association, you make the inference that these are disease-associated variants. These are all very good questions. Cool, guys. All right, so then we're done. Thank you.